Hello everyone. It's great to see you. We have a steady flow of attendance every week or twice every week. That's absolutely great. So today is kind of exciting for me to welcome Hadi Valadi, who used to work in my lab back more than 10 years ago now. And Hadi, together with Karin Ekström, were first authors on the paper that we published in 2007 that described the shuttling of RNA from one cell to another cell through exosomes. So Hadi, after that, got a nice grant and developed his own independent lab. So he's now an associate professor in Department of Rheumatology, right?
Yes.
He's been working for several years as well with Nawaz Mohammed, who is a senior postdoctoral researcher, has been working with EVs for a number of years now, and working closely with Hadi. So Hadi, it's a great pleasure to see you here, and we're looking forward to your presentation.
Thank you very much, Jan. Thank you very much. So it was very nice introduction. Yes. So the idea is that actually I will present our research interest first, and then afterwards, Nawaz will present our recent publication. So our research interest is about to explore how cells send different RNA molecules to each other via EVs, and how to utilize this process for therapeutic use. As Jan said, actually, in 2007, we find out that the EVs contain substantial amount of the RNA, and we could show that they contain both mRNA and macroRNA. And since then, it has been published many papers showing that EVs contain RNA and they are capable of delivering this RNA to other cells. But all these publications together, they have something in common. They show that actually the RNA in exosomes and cells are differently expressed. So it means that if the RNA in exosomes are differently expressed compared to the parental cells, it means that there must be a sorting and loading mechanism. A mechanism that sorts some of the cytosolic RNA into the EVs during their biosynthesis. So first, since RNA binding proteins bind to all the RNA in nature, there is no RNA in nature that is naked. All the RNA in nature, from plants to bacteria to body cells, they are always complex with proteins, such as, or so-called RNA binding proteins. These proteins, RNA binding proteins, they determine the fate of all the RNA, such as translation, translocation to different cellular compartments, or degradation. So first, we hypothesized that maybe there are RNA binding proteins that can select which RNA should be loaded into the EVs. Therefore, we search for the presence of RNA binding proteins in EVs. Using gel shift, we show that EVs contain RNA binding proteins, and using the affinity of binding of RNA between RNA binding protein and RNA, we could identify 30 different RNA binding protein in EVs. And by silencing some of these chosen RNA binding protein present in EVs, we could show that some of them are responsible for loading of the RNA into the EVs. And then we went on, and then we want to investigate whether exogenous mRNA that cells taken up have the same fate as endogenous mRNA. So using lipid nanoparticles, we deliver a functional mRNA encoding human erythropoietin to cells, and then we search for or we investigated the fate of this mRNA in cells, which Dr. Nawaz will tell you more about it. And as Jan said, Dr. Nawaz is very experienced EV researcher and has been in my group more than 4 years. And he has been very much involved in this project. Please, Nawaz, you can continue.
So thank you very much for the nice introduction, both from Karina, John Lotvall, and Hadi for the kind words. So as Hadi already has briefly informed about our recent paper that was published in Nature Communications. So we have created one linkage between the lipid nanoparticles when they are endocytosed, and then they are loaded into vesicles, this messenger RNA from lipid nanoparticles, and then we deliver this messenger RNA to other cells. So briefly about our lab, we have been for 3-4 postdocs, Marco, Alex, me, and there were also some other master students, but I have only few pictures available, so I'm showing. So we also have a Twitter account from our lab, Valadi lab, if you want to follow. So then before presenting by our paper, briefly I want to share with EV community, because I think it's my first ever presentation with the this community. So I want to present myself from where I came. So I did my first entry into academia was 2004, GC University Lahore, and it's a more British type of educational environment. And then I came to know that there was a Nobel laureate, her Gobind Khorana, and he born in Pakistan, but later Pakistan and India was separated. And why it became my interest, because we are studying the nucleotides and the genetic codes and protein synthesis, and it was of very interest for me. And it was graduated from the same university from where I was. Then he left for USA, and later he got the. So then there are a lot of mentors who had a very strong impact on my academics. And first I started my master thesis at Quaid-e-Azam QAU, Pakistan. And my mentor was, he also did a postdoc from Baylor College Medicine, USA. And I learned from him genetics, genomics, genotyping, and also good, molecular biology. And then Wasim Ahmed, and he was also a good name in our university in terms of genetics, genetic population. And he did his PhD from Cambridge, postdoc from Columbia, and I learned nucleic acid and protein chemistry. And then I briefly tell a few names that they were, why I'm saying that I have been mentored by people or professors who were graduated from university from USA, UK, and abroad. So they had I was already living a kind of in an international environment. So then my PhD supervisor, he was also graduated from Harvard, and he did postdoc and from Mayo Clinic and Harvard. And then I was introduced to Hadi. Then my life changed. Actually, I entered into exosome field, and he was my co-supervisor. And then I continued postdoc, and then continued in the lab. So what I'm doing now, let's go to the exosomes. Actually, I collect a cellular trash and make it a trayer. And with the apologies with the EV fields, I'm not saying the EVs are the trash. It's just symbolic. And it's a mimicry. So actually the EVs have a lot of potential. They have a lot of potential to be explored. So we are exploring this kind of their trayer potential. So then what is, you see that from outside and what actually they do. So we are observing them, their surface presentation, let's say CD63, CD9, they look very cool, but what actually they do, their work is to do a cargo delivery. So what we are doing now. So what was the main reason I came to Hadi Valadi lab? This was the paper. It made me very, it brought my interest to this field. So I studied this and I contacted Hadi for PhD exchange, and then I got to Gothenburg. And then I was also using, in my project, I was using some liposomes for delivery to glioma cells. So then I used that, I thought that exosome could also be used as delivery vectors. So then I read this paper that Hadi introduced one method, that electroporation, that there were other groups also use this method, and this was my interest. So then we come to our, without wasting time, so we come to our main paper that we recently published. Then we used the nanoparticles, and these nanoparticles contain messenger RNA, and they were delivered to cells, and we harvested the exosomes, and we found that this messenger RNA was in the exosome from the LNPs. So these are clinically advanced delivery, the most advanced delivery system to date are the LNPs, and they have been used in to delivery for the siRNA, messenger RNA, and the co-delivery of the pod. And then they have also been used in clinical trials for influenza, Zika, Ebola, and recently SARS-CoV. They are also using these LNPs. So then briefly the structure of ionizable, why I am showing this structure, because this we will, in my paper that I present, we will focus on these amino ionizable lipids and the nucleic acid, that is messenger RNA. But they also have other components. So this is they mimic some way in vesicles in terms of their size and surface and delivery. So LNPs have been very efficient for their endocytosis. There are a lot of studies that have shown that LNPs can be endocytosed, and they are found in the endosomal system. And there were very interesting thing that within the first half hour, most of the LNPs, about 95% of LNPs are endocytosed. So it means they are inside the cells. So, but when they are inside the cells, what is they need to be released into the cytoplasm to show the action. But there is a problem that there is a limitation, what limits. So they have a limited capacity to go to endosomal escape. So they are stuck in the endosomal pathway. And it has been shown in several publications that less than 2% of LNPs that were endocytosed, less than 2% can escape from the endosomes to the cytoplasm. So what it means, the 98%, let's to be on safe side, 95, 90% of endocytes LNPs, we don't know what happens with them. So previous studies have shown that maybe they are degraded, maybe they go to autophagy, lysosomal pathway. But we thought that let's explore this. What is their fate? So Hadi came up with the idea that why not we should see in the exosomes, because the previous studies have shown that they are endocytosed, they are in the endosomes, but no one has studied their exocytosis, especially we explore in that. So what we did, we delivered a one messenger RNA that encodes, it's an APO, it's a erythropoietin, that encodes the erythropoietin, and that protein is a secretory protein that has been used in treating the blood disorders. So we delivered first, we delivered the APO messenger RNA to cells via LNPs. And what we see that, as the previous studies have also shown, that they are endocytosed and early endosomes, and less than 2%, as I said, so less than 1% in our case, is escaped. The part that is escaped here is also functional. So, but it's very little. So functional means that a messenger RNA is translated into apoprotein. And as I said, that is a secretory protein. so that can also be secreted. So first we identified, so we in the cell lysate, we collected the lysates and the isolated RNA, and we found that this RNA was found in the lysate. So we used two types of different LNPs, lipid nanoparticles. There is the difference is based on their fatty acid length or their composition or formation. So we named that DDLNPs and MC3. I have definitions. One has a D-lin MC3 ionized lipid, and other has a D-lin DMA ionized lipid. So when we change their lipid composition and we see their delivery of apoRNA, one versus other is significantly different. So MC3 was very efficient in terms of delivery, but still it's less than 1%. But still we were curious to know, we will present that what we did. So then we also checked that, okay, messenger RNA is there. Can we see the protein? And the answer was yes, we did ELISA. So we could see the protein. Then we further said, okay, this protein is secretory protein. Is it also present in the supernatant? It's secreted from the cell? And the answer was yes. So in all the cases, MC3 was giving us kind of good results, so comparatively significantly higher. So this was the, again, that less than 2% is escaped. What happens to the rest of the messenger RNA? So now we came our hypothesis that when it's endocytosed, okay, this part we have already discussed, but now we will go to the other part. So from the endosome, when there anything is foreign particles in the endosome, it has a relation with the multiple vesicle bodies and exocytosis. So we were curious to know that if we could find this messenger RNA that was endocytosed from the LNPs and whether it goes to the exocytosis. And then here we will use this endo-EVs term, that is the EVs that were derived from the endocytosis of LNPs. So then we isolated these vesicles and we checked if there is a messenger RNA, and the answer was yes. We did qPCR on EVs and we detected a good amount of this messenger RNA in apomessenger RNA in EVs. So here again, these MC3 LNPs were giving more in EVs. So exocytosis from this type of LNP was higher. So then we characterize these EVs to show that whether they are the size, and then there should be the EV size, and then we did also RNase treatment, whether the messenger RNA is really inside the EVs or it was just stuck from outside the vesicles. So this was a good question we discussed, and then we treated with RNase, and we see when it was treated with RNase, there was slight decrease. It was, then we concluded that this small portion, maybe there is, although there is a chances that some of the part has been unspecifically outside, but majority of the RNA, messenger RNA, was inside the EVs. So this gave us a confidence that the RNA is inside the vesicles, and the messenger RNA especially is inside the vesicles and is protected. So then further we went to see that, okay, if we capture these vesicles with CD63 beads, and then we analyze this messenger RNA to see that whether these CD63 positive EVs could also have messenger RNA, then we used one label messenger RNA, Sci-Fi messenger RNA, and we see that this population of vesicles that were isolated by CD63 capture, when we conjugated them with CD9, and we see that 96% of these vesicles were positive for CD9. then, okay, these vesicles are positive for cDNA. Do they also have messenger RNA? Then here we see that yes, they were positive for this messenger RNA. What it means that the vesicles that were positive for their typical markers were also positive for their messenger RNAs in them. So then we had a lot of discussions, and I will keep asking these questions to ourselves, and as we have been discussing. So next question was that, is this only because at the beginning I said that messenger, these LNPs have other components also. So was that only messenger RNA inside the vesicles, or the other components can also be incorporated? So the original LNPs that were delivered to the cells contained 3 moles of ionizable lipid that we say that here, ionizable lipid, cholesterol, the SPC, messenger RNA, PEG lipid. So out of when we delivered this, so they had a 3 mole of ionizable lipid per messenger RNA nucleotide, not the whole messenger RNA, per messenger RNA nucleotide. So the ratio was 3:1. What was very fascinating to know that this ratio, when we identify the same ratio in the vesicles, it was neutral. And we thought that could be one reason that if they have been processed inside the endocytic pathway, and some of the ionizable lipid has been processed, and we see that this ionizable lipid and messenger RNA ratio was 1:1 in EVs that were secreted from MC3 lipids, and the ones that were DD. So in both cases, no matter which type of LNP we used, MC3 or DD, in both cases, the ratio was the same. And we kept on doing these experiments one after other, and we did 13 times. And the experiment repeated 30 times, and all the times, although values were a little bit up and down, but the ratio was always 1:1. And did give us a very confidence that there is some reason. So then Hadi came up with a very good hypothesis that we will discuss at the end. Just to remind you, keep this eye focused while I explain this ratio. We will describe one mechanism at the end of the presentation. So then we had one question that, okay, so we found this some part of ionizable lipid in the EVs. Was that incorporated from outside the cell culture, or was it from the endosome? So then we see that the relation between ionizable lipid and messenger RNA remains the 3:1. So if it was contaminated, or it should have like incorporated with EVs from outside, so the ratio should have always 3:1, because it cannot be processed. So this ionizable lipid and messenger RNA ratio is always intact. So if they are have contamination, they should have this. But we see that the ratio was not this, it was 1:1. And this was a kind of another confirmation that it has been processed. Otherwise, it should have been 3:1. So then further we asked that whether the interaction occurs between LNPs and EVs outside the cell culture when we don't have the cells. So we mixed the LNPs with the EVs that were not treated with LNPs. And then we incubated them for a time. And we could see that there was no messenger RNA found in these EVs then when we mixed them with the LNPs. So when we compare with the other ones that were treated with the cells, the EVs derived from the LNP treated cells contain messenger RNA. But when these EVs were mixed with LNPs with those cells, there were no RNA. So then further we confirm that it's not happening outside cells, it's happening through the endocytic pathway. And also a lot of studies have shown already that LNPs are endocytosed. So then next question was that, okay, we have found this messenger RNA, we have characterized, now we are sure that they are inside and they are protected. So can they deliver? Is this functional? So can they deliver to the cells and they can produce a protein? So then we used a label messenger RNA to track there, whether it's inside the cells. So we used epithelial. First, we delivered this labeled messenger RNA via EVs to the epithelial cell line, and we could see that these cells were taking up these EVs, and there was a significant amount of, almost 60% of these population contained these EV messenger RNA. And then we went to the human peripheral blood cells, PBMCs, and we used the FACS analysis on B cells, T cells, and monocytes. And why we use this? Because it has been shown that the immune cells, especially the B cells and T cells, they are hard to transfect with messenger RNA. And we studied literature, and we couldn't find that there are studies that could transfect these cells with RNA delivery to them, especially the messenger RNA delivery. So then in our case, so we found that like B cells and T cells and monocytes were positive for our messenger RNA that we deliver. So then we said, okay, this was a labeled messenger RNA. What about our functional APO messenger RNA that we have been studying? Does it do the same? So yes. So when we delivered this and we checked the protein production, so it means this protein is coming from the APO messenger RNA. And then we did a control experiment that whether these cells already don't have this erythroprotein. If they already have, then it will be like how much they already have and how much has been delivered by EV. So we did a control experiment, and they didn't have. So when we delivered this messenger RNA, and we could see that apoprotein was in the cells and were also in the supernatant, and we confirmed that yes, these EVs can deliver functional messenger RNA to produce apoprotein. Okay, so next question was, as I said, you will see a lot of questions and their answers. So next question was, we had hot debates that, okay, let's see that maybe in vitro and in vivo environment, in vivo is more important. Do they do the same in vivo? So if we deliver them, these vesicles containing messenger RNA to the mice, and can they produce the apoprotein? And we delivered the messenger RNA via EVs. And we did intravenous injections, and 1.5 microgram of messenger RNA was delivered. And then we did a micro sampling. There was a device that collects a very minute amount, and we followed, the time course for 2 hours, 5 hours, 24, 48, and 96. So we collected, and we could see when we did perform the ELISA, we could see that messenger RNA has produced the apoprotein that was found in the blood. So as I mentioned earlier, because this protein is a secretory protein and is used in blood disorders, so it's obvious that it will be found in the blood if messenger RNA was functional. So it gave us confirmation that yes, the messenger RNA has delivered, EVs have delivered the messenger RNA, and this was functional, that can be produced and secreted. Then we asked, what about the organs? After the organs, we had 8 organs. So in 8 organs, we detected, out of 8, we detected in 4 of them. And it was both the EVs. I can use this slide, but we could see this protein in heart, lung, liver, spleen, and also the blood. So out of 8 organs, we could detect in both the protein and messenger RNA in these organs. And but these 4 organs were negative, like kidney, thymus, and pancreas, and brain. So we could not detect in them. So then we compare them with the LNPs when we deliver the same amount of messenger RNA to mice with the lipid nanoparticles and the EVs. So we could see that LNPs deliver comparatively more than EVs. There was then we thought that maybe these there are some of EVs don't have messenger RNA, or we need to improve our method to incorporate more messenger RNA to deliver efficiently. So, but what we can conclude from here that these vesicles are that were harvested from the endocytosis of LNPs contain the messenger RNA, and they can be delivered to cells in vitro and in vivo, and they can produce the protein. And oh, actually, sorry, I actually the sequence was a little bit disturbed. So now here I can show you. I was a little bit nervous also that that how it's possible it's missed. So sorry for that. So here we see that the spleen, liver, and lung. We tested both this panel shows the messenger RNA, and this panel shows the protein. So we could see that in both cases, in terms of messenger RNA delivery via EVs, we could see in the organs and also the protein production. And the spleen was the organ that shows the highest amount of messenger RNA, and the liver was the one that has the highest amount of the protein. So these two were showing comparatively higher amounts, but these all four were positive. So, okay, so then we did a one control experiment. If we are doing this ELISA on human APO, maybe the mice has also this human version of APO, or our kit can detect that. So we checked cross-reactivity with the human APO versus mice APO. So it was a very good indication for us that when we performed the human ELISA, human APO ELISA on mice APO, and we could not see any signals. So here we can see that there was no signal. So then we were sure that because the mice lack this APO, so what we see there is the human APO. So that was one other control experiment that gave us a confidence that what we are delivering and what we are getting in mice is the human protein. I mean, newly messenger RNA and new protein. So then we checked, okay, LNPs deliver a little comparatively more than EVs, but what about their effect on inflammatory cytokines? So then we checked that messenger, when we deliver the same amount of messenger RNA via LNPs and EVs. So EVs were, if we see for example, here interleukin 6, IL-6, so EVs had a less pro-inflammatory response than LNPs. Also in case of IP10, and the LNPs were showing higher inflammatory response. And in case of these all, we can see that EVs are significantly less. So especially in these cases. So then we said that, okay, so EVs can be a good choice for delivering the messenger RNA than LNPs, because LNPs are synthetic, they have the lipids. EVs are kind of natural, and we need to improve the messenger RNA delivery, and that could be a good ideal candidate. So as we have been discussing of our ratio, if you remember in initial slides, that the ratio between ionizable lipid that we originally deliver contain 1 mole of messenger RNA per 3 mole of ionizable lipids. When it is endocytosed, and actually we detected 1-to-1 in EVs, if you remember, just recall the slides previously. So we will connect this 1-to-3 and 1-to-1 in this mechanism. So when they are endocytosed, and it is a very established fact, they are going to early endosomes, and endosomes can fuse with the lysosomes, and the lysosomes have the acidic pH, and when they fuse with the acidic environment, so there is an acidification step. So what it means, then the pH is lower, acidified. So it is highly possible, or kind of surely possible, that LNPs have been positively charged. And we know that the endosomal membrane is negatively charged. So LNPs, naturally their surface is positively charged, but when they get more positive charge by acidification, So their interaction with the negatively charged endosomal membrane becomes higher. So then how then their fusion occurs, and when they need to be escape, they need to be neutralized. So that was our point here. So they need to be neutralized into 1-to-1. So the messenger RNA and lipid ratio should be neutralized when they need to be escaped. So when we talk of endosomal escape, it can be of two forms. One that we have been showing that a part of that is escaped into the cytoplasm. That's the messenger RNA that have been showing that less than 2% is escaped, and the messenger RNA can be translated into apoprotein. Then there is an other form that can go to the into the, you see this endosomal membrane. It has invagination has occurred, and then this lipids, LNPs inside the endosome, and then we have the messenger RNA can go to the intraluminal vesicles of the endosomes. So this needs to be neutrally charged to come from the endosomal membrane towards to the cytoplasmic membrane, because the 3-to-1 is highly positively charged, strongly positively charged, and negatively charged. So it needs to become a salt, neutrally charged salt, so that it can escape from the endosome and it can be packaged to the interluminal vesicles, and it can be secreted. And it was confirmed not only by this ratio, but we also could see, we did a 1 pH experiment. Here we can see that when we see the pH is acidic, and the release was less. So then we increased the pH and the release was a little bit higher. When we further went to the kind of neutral, so it means that it needs to be neutralized. At neutral pH, there was more dissociation and release. So it was kind of a confirmation that this 1-to-1 ratio is very important. And we did it, as I mentioned, 13 times. So in different form of LNPs. So results were always the same. So it means there is a pattern. The cells are showing some pattern. They are showing some a consistent mechanism. So there is a purpose. So then we said, okay, this was in the mice and this one, because we see that these LNPs have been advanced to the human clinical trials, and a lot of companies, I will not mention the names because I am not here to promote the companies. I am an academic guy. So actually, on a serious note, that they have been used in clinical trials. What is happening in clinical trials, that LNPs are delivered to the human and they are packaged into EVs through the same mechanism that we shown, that they are incorporated in the EVs, and these EVs are delivering to other cells. What it means, that the part of RNA that we see in the cells can be from EVs instead of LNPs. So part of LNPs is delivering messenger RNA, and the rest is happening. We don't know how much is, but it's a hypothesis that it can happen because there is a established fact that LNPs are endocytosed. When they are endocytosed in the human, they can also be packaged into endo-EVs and they can deliver to other. So we are very, very excited about this in future if it has been discovered some way. So this was our kind of confirmation from our mice experiment and in vitro experiments. So then we published this paper in Nature Communications, and Marco Maugeri, he is not here, he's the first author, and we had a good time and a good experimental, and we enjoyed actually for 4 years, and a very strong mentorship and hot debates, very hot questions we had, and then we figured out the new ways that there is a link between endocytosis of lipid nanoparticles and their escape from the endosomes and their loading into vesicles and delivery to other cells and production of protein from this messenger RNA. So that's it. So then I would definitely acknowledge all the friends and our lab mentor and the funding agencies and the University of Gothenburg, and the project was funded by Vinnova. Vinnova is Sweden's innovation agency. Then is Sweden's Strategic Research Foundation, and the project was also conducted in collaboration with AstraZeneca. And yes, then two of these hard times, my family have been with me, and they were always giving me positive energy. My child, my wife, and this is very important when you are in a tough time. So to be. And thank you, of course, for your patience. And now we can go to.
Is that Hadi sleeping? I'm just joking.
Maybe I was thinking I'm only the one that talking. Everyone is sleeping because I cannot hear. Yeah,
no, not
muted.
at all.
They are muted.
Thank
You see
so
that
much.
the administrator is muting them. So
yes.
So here we go. I stop sharing. Yeah.
So everyone can ask questions, and by posting them on the chat box, and then we will unmute yourself when you ask the question. So yes, Leti, you put the question in there, but Leti, ask the question, unmute yourself and ask the question.
Hi, I'm Ming. I'm calling from Singapore. I wonder, first of all, this is a very nice talk, and it's my favorite topic. Very nice to see the work being published. And I wonder what the proportion of mRNA you transfer into the cell using LNP go into the exosomes. Because you mentioned that there's only the 2% that escape from the endosome, and you assume that only the mRNA that have escaped can go into exosome, right? So is that 2% of the starting materials, or you have much less than that? So is it an efficient way of delivery and getting exosome with this mRNA? And the second question is, what type of cells do you find the mRNA in the organs? Because most of the LNP go to macrophage, especially in the liver. So is it better when you have exosome to carry this mRNA? Do they go to other cell types that are more meaningful?
So that was three questions, right?
Yeah. So coming to your first question, that when we say that less than 2% is escaped, and it means let's say 98%, or this 98%, it doesn't mean that it's all of it's going to vesicles. So in the figure, there was a percentage, it's less than 2% also in vesicles, but it's not that still a lot of it's degraded. It goes to lysosomal pathway, autophagy. But the thing is that the proportion we have definitely, we delivered, let's say 1 microgram of administered. So we did a percentage versus administered messenger RNA, that we administered this much, we got this much in the cytoplasm, and then we got this much in the EVs. It was also less than 2%. So still it's about 4, 5, 6%. And we don't know what is happening remaining 80, 85, 90%. So, but the good thing that we are positive about that, it is incorporated in vesicles. And as we said that if there is this pH thing, or we can engineer the cells in a way, or EVs to incorporate a higher amount of this messenger RNA into vesicles. Then coming to your next questions.
Can we stay with that question for a little bit? So other people have reported that LNPs are more efficient in delivering cargo to cells. And if I understand correctly, the hypothesis is that they have a tendency of fusing to the membrane at the surface of the cell, whereas extracellular vesicles or exosomes are taken up through the endosomal pathway, and a lot of that material ends up in the lysosome. So could you comment on that?
I don't know if Hadi want to say, or I have my own version, but Hadi, if Hadi has his version.
Yeah,
I mean, yes, of course. I mean,
both.
it has been, yeah, it has been published actually many times that LNPs are taken up by cells via endocytosis. I mean, they form endosomes, and there are macroscopic pictures. There are many ways they have shown that LNPs is inside the endosome. But what is inside the endosome doesn't have any function. I mean, nothing has function inside the endosome. It means that actually to have a function, it has to escape, the material has to escape from the endosomes and end up in the cytosol. Because in cytosol, the mRNA or siRNA is functional, can give rise to the production of the protein or silencing. Inside the endosome, nothing happens. So this is the very important part of RNA delivery via LNPs, that it has to escape from the endosomes. And when it escapes, I mean, how it escapes, nobody knows exactly. But we claim that the RNA must be neutral to be able to pass through the membrane of the endosomes and end up in the cytosol. Then it's functional. And when it passes through the membrane and end up in the cytosol of the cell, then of course part of the RNA can go inside the luminal vesicles of multivesicular bodies, late endosomes, and secrete like exosomes. I don't know,
Nawaz,
Jan,
what's your version?
Yeah, actually, this they are like LNPs have been showing their high the endocytosis, but still their efficiency is not, even though if they are 95% are uptaken, but they are not showing they are not showing that level of function. I mean, they are endocytosed, but 95% or 98% is not in the cytoplasm. That needs to be in the cytoplasm to show the function. If it's siRNA, it needs to be in the cytoplasm to silence. If it's messenger RNA, it needs to be cytosol to be produced, translated into proteins. But although they are, what my point is, although they are endocytosed efficiently, all 95% of them, but they are still, the functionality is limited. Versus EVs, EVs have we are like after several years of study, we are still not sure about a lot of questions that we need to answer, that we need to improve these EVs for the delivery, for the uptake, and for more to be more targeted. Like Lead said that which cell we have identified, and we did not perform at cellular level, we performed at organ level, and still we could see that the spleen and the liver were the highest. So then the further which cells that we did not dig up that.
Thank you very much, but I'm seeing a bit confused. So do you mean that the mRNA that go to the exosome is from the 2% that escape the endosome, or is from the 98% that
no, no, it's not like this. Actually, previous studies have shown this. They have shown that they are not clear what happens to the 98%, the remaining 98%. We show that let's figure out where is this 98%. So then still out of those 98%, some of it we could identify in vesicles. So it's not that of 2% escaped. 2% escapes in the cytoplasm, but then there's also escaped that goes, escape from endosome is not only the cytoplasm. It has been escaped from the endosomes. Either it has gone to the cytoplasm or it has packaged to the vesicles. So the previous studies has shown that the escape is less than 2%, but they have not shown that this escape is also linked to the exocytosis. So then part of this escape, we also detected some of this. So it's not all out of that 2%. No, it's out of that has been endocytosed. So out of endocytosed, what we get in EVs? Yeah, it was less than 2%.
Oh,
Also.
it's very, I'm sorry interrupting. It is very difficult to know, because when the RNA escape from the endosomes, how much of it exactly translated go through the dissociation, and how much of it goes exactly into the luminal vesicles of endosomes is very difficult to answer. So we don't want to actually say exactly any. But we say that we found part of the RNA inside the EVs, but exactly how much, I think nobody will be able to answer it. To your second question, is very good question actually. Which cell types? Of course, macrophages are very interesting. Macrophages from the liver, but we didn't look at it. So we cut actually a piece of the organ, and we search for the RNA, mRNA, in that organ, because we needed to be sure that the mRNA is there as well as the protein. Otherwise, we couldn't actually decide that the protein was delivered by blood to that organ. So we needed to detect the RNA also in certain organ. So it was a good question. Thank you.
Thank you very much.
Okay, so we go to Genevieve Bart. Please unmute yourself and ask a question.
I haven't. Sorry. I'm from University of Oulu, Finland. I'm actually curious about the delivery of the EVs in animals, because I have seen most reports say that it goes to liver and spleen. And I was just wondering if you had not thought of kind of a more physiological ways to get these EVs, because most EVs would be secreted by cells. They would be in interstitial tissue. So most of them, would they really end up in blood and then in liver and spleen, or? I don't know if you have any.
I understood that. Actually, we did not identify or investigated in the interstitial spaces or in the secreted. Then it will be a secondary vesicle that the EVs deliver, and then they are secreted. So it becomes a secondary vesicle, a secondary vehicle, or tertiary vehicle. So we have not investigated that.
So
what we found,
So actually, the follow-up would be: have you injected your RNA in the lipid nanoparticle in animals?
Yes, we did, and we also compared. I showed in a figure that we injected the same amount of messenger RNA via LNPs, and then we use the same amount of messenger RNA via EVs. So then we compared their production of both the protein and secretory proteins, and then we also compared the inflammatory responses.
But that you check. Then you check the EVs in blood, right? After
the production of when we deliver EVs, messenger RNA via EVs, then we check the production of protein, because this protein is a protein.
This part. I'm just asking about the animal study. So when you inject your lipid encapsulated mRNA in blood in the animal, and then do you check the EVs in blood, or did you not do that?
No, the messenger RNA was not checked in the blood. But yes, not messenger RNA, but the protein, of course. Okay. Because the protein is produced from the cells and is secreted. It's a secretory protein.
Okay. So, now I understand.
Yeah. Okay, to straight away to your question, we didn't check the messenger RNA in the blood. after delivery of LNPs. We check the protein. Yeah.
So
Okay.
Yuval De Beer has a question, and he has a non-functioning microphone. So does the in vivo loading work with siRNA transfer for LNP to EVs as well? So if you put siRNAs in there, will they go to the EVs as well?
The studies have shown, yeah, okay, Hadi, yeah?
Actually, it's a very good question. So we are wondering also, so if the similar way, siRNA will end up in the EVs also. But the question, I mean, we don't know. To answer this question, yes, we don't know today. But we have to remember that siRNA is double-stranded. And this double-stranded pass through the membrane of endosomes and end up in the cytosol part might be tricky. But if I guess, I would say yes. But the answer is we don't know, Jan.
Okay. So he's also asking, following up, that when you look at put LNPs on cells, they're taken up by the cells. Not all of them, but some of them are taken up by the cells. Then they're excreted again. How do you separate the LNPs remaining in the supernatant from the exosomes? I think that's basically the question.
Yeah, this is a very, very good question also. Yeah. So I think actually Nawaz answered very nicely actually during his presentation that we did our best to show that actually our EVs are free from the LNP. I mean, what is known, it has been shown in many, many publications that 95% of the LNPs are taken up by cells during the first 5 hours. We isolated our EVs after 4 days. And at the same time, when we isolate them, we fetch them by antibody, CD63 antibody, and then to be able to get only EVs, not the LNPs, as well as actually we RNA treated. We did really our best to show that our EVs are free from the LNPs.
But we also mixed them with the EVs without the cells, so we couldn't see the direct transfer.
But to answer to this question, I would say that maybe there are very few contamination, but we couldn't show that. But maybe there are. When we mix actually EVs with the LNPs outside the cell in a cell-free environment, right away we saw that actually aggregates such a big particles. It means that actually one LNP try to fuse with the EVs, but it cannot enter. And then another EVs and another EVs and another LNPs, then you get actually such a big particles. So what I believe that it happens also outside the cell. If LNP will fuse with the EVs, is not really possible. It make aggregates, it make big particles.
Okay, so we move to Michael Rimmer, please. Unmute yourself and ask your question.
Hi there, my name is Mike, I'm from Scotland. Thanks for a great talk. At the very beginning, you briefly discussed how preferential loading of the RNAs into the EVs may occur through certain proteins which help to load them, and then you've very nicely demonstrated how the presence of the RNAs in the endosome is what's required. Do you think there's anything else happening in the endosome? Are there other molecules that will preferentially load these RNAs? I'm just thinking in situations where maybe you've got cellular stress or you're treating the cells with chemotherapy, that would induce other pathways. Do you think that will influence the RNA loading from the endosome if you were to incorporate specific RNAs before treatment?
Yeah, actually, if what we, as Hadi initially presented the role of RNA binding proteins, there can be molecules that are preferentially sorted. But we are studying only one messenger RNA, but there are a lot of other cytosolic RNAs, microRNAs, other non-coding RNAs. and their sorting happens through a control mechanism? And the answer is yes, happens. But we in case of messenger RNA, we didn't this messenger RNA, we didn't check other players that are important, that RNA binding proteins or something. But there could be, it cannot be excluded. So there is possibility that it can be controlled. But we have shown this ratio. They should be neutrally charged to escape and packaged into the vesicles.
Okay, thank you. I missed Aaron Scott. So Aaron, please unmute yourself and ask your question.
Yeah, so a great talk, Nawaz. It was really great, thank you. And I guess my question really was linked to what a few of the other questions, wondering about the different cell types that might be taking up these EVs. But then I just wondered, with your data on the inflammatory cytokines, whether this suggested maybe different cell types. So you saw that there was less of an inflammatory response from the EVs compared to the LNPs, and then whether this might suggest that there's a difference in the cell types that are taking up.
Yeah, cell type, or yes, because we used one type of mice. It was a bulb C. So we don't know what happens to the other mice. Like if you go to the different models or different doses or different. So we used only one mice, but which cell types? And we didn't, as someone also asked, that we didn't go to the cellular level in the recipient organs. So it can vary. So it can vary cell to cell type. And also in vitro, we used epithelial cells and PBMCs, peripheral blood. But if we go to other cells, we have not checked the other cell lines.
I would like actually to comment on this too. Thank you, Aaron, for your question. But I mean, is it not the same question actually? Everybody of us who work with the EVs actually struggling to figure out how to target it to a certain cell type. I mean, there are some publications, but to be honest, all of us, we have difficulties to deliver it to a certain cell type. I don't know what Jan or what yourself actually would like to comment on targeting.
I wonder. So yeah, I see the same with my own work as well. So when we inject EVs into zebrafish, we see preferentially that they're ending up in macrophages, sometimes endothelial cells. But I just wondered, from the cytokine expression data that you had, where it looked as though the EVs were inducing less of an inflammatory response, whether that might suggest that you are somehow circumventing that problem, if it's a problem at all in the first place. Yeah, it was more to do with that data that I was wondering if you'd managed to avoid that bias.
But have you succeeded actually to do any targeting, or anyone can talk about? Or...
Yeah, we've discussed that before in these seminars, that so there was a paper in 2013, and I was fortunate to be co-author on that from Young-Sung Go's lab in Korea, where they made vesicles. They actually manufactured vesicles from inflammatory cells. And if you activate the endothelium, you express adhesion molecules on the endothelium, and then you have your matching adhesion molecules on the vesicles that you make. And then there is a preferential uptake to activated endothelium. But that's the kind of with intravenous injection, right? Otherwise, you would have to look quite carefully into specific uptake mechanism. You have to pass over the endothelium if you inject intravenously. You have to pass over the endothelium to get into a cell, then you get into the cell, and then you get into certain compartment inside of the cell. So that's kind of interesting and not so kind of complicated.
Yeah, it is. We notice also.
But that said, you can probably engineer for preferential uptake in certain cells, and we just need to understand a little bit better how subpopulations and so on. Before
then the question comes to the immune cells, because our question,
and
yeah,
dendritic cells and macrophages are very, very prone to take it. They're basically exosome eaters, aren't they? I mean, before we go to the last question, I wanted to comment on a thing that Hadi said, that vesicles are inactive in endosomes, and and from the delivery perspective, you might be right. But we just published a paper about a year ago now from Ganesh that there seems to be signaling, surface receptor signaling, TGF-beta receptor signaling inside of endosomes. We had a lot of data. There's some other published data that implies that you can have receptor activation inside of the endosome. But
yeah,
delivery,
but we have to.
Yeah.
Yeah, but we have to actually remember that actually the role of lysosomes when they fuse with the endosome is that actually to degrade the content of, yeah.
That would be before that happens, actually.
Yeah, exactly. Maybe it is. Yes. Yes.
So, Fam, could you unmute yourself and ask your question, please? So this is what about organ harvesting, and how that, when do you do that, and so on. So timelines, I think.
So the harvesting of organs or the blood. Blood, we did. There was a device for micro collection.
I think we could extend the question a little bit and think about kinetics. How fast does it happen? How long does it stay?
Actually, yes, we have shown the time course from 2 hours to 96 hours, but we could see between 2 hours to 24 hours. And in some cases, if we go to our one of my slides.
And if you do repeat injections, can you prolong and extend?
We did not perform the repeated doses, time lapse dose, what you said. So we did not do that. It was one single dose, but we didn't follow up the multiple doses. So it can be like, then in this case, it needs to be repeated every 24 hours because it stays. Right. Then because this process is not a static, it's a dynamic, that machinery is being uptaken and it's being translated into proteins. So which is the peak? Then we need to follow up the times and multiple doses. But in our case, we checked, 2 to 24
Now we have
hours.
Tien unmuted. Did you get a response to your question?
Yeah, thank you very much for the talk. Yeah, I think I'm satisfied with that.
Thank you very much, both of you, Nawaz and Hadi. Big hand from us, everyone.
Thank you.
Thank you, everyone, for the great discussion. And I'm handing over to Carolina to sum up end the meeting.
Yeah, thank you so much, Hadi and Nawaz, for a great talk. So I think there's a lot of things was discussed about how we can deliver mRNA and what is the process that is happening between the LNP being transferred to the multivesicular bodies and being exocytosed in extracellular vesicles. I think it's also quite exciting that some very, very less mRNAs actually escape and being translated in the first cellular target. So what is actually the comparison, I guess, in the future between the LNP and endo-EV will be quite exciting in terms of the functions. I think that's to summarize for today's talks, and I hope to see everybody next week.
Thank you very much.
Thank you very much, Jan, for question and answer. Sorry.
Bye.
Thank you.
Bye.
Bye. Bye.